How do I actually use the system? Everything is done through the software. As you saw in the previous images of the Hermes, there are no oculars, so you don't have any manual adjustment. Everything is done through the software control. We've tried to make it as intuitive as possible, and if you used other software, you'd probably be able to figure it out fairly rapidly. So the first step is to select the application that you want. The most general one is called this image acquisition application. It opens up all of the different colors, all of the different magnifications that you have put inside. If you have a specific assay that you want, for example, you only want to count cells in one color, then these other applications are streamlining the whole image acquisition and analysis process, but it's not required to choose any other application. Just start with image acquisition. Then select the objective that you would like. You have the first objective. If you have the rare event application selected, the objective number 2 will also become activated. Then you would select the colors that you want. So they would be the bright field for gray, blue, green, red, and far red. Then scan setup. So we'll enter into this menu. And here you're telling the software, where do you want to image? So we have a predefined list of multi-well plate and sample types in this upper corner. So you would choose the sample type or the plate that you're currently using. If it's not present, just let Arcadi know, and we can support the incorporation of your custom plate using the data sheet, right? So once you choose the plate type, you'll get the layout shown here. Then we tell the software where to image. There's these two parameters, well coverage and field density, and you'll get a readout right here for what that means. So this outer circle shows the edge of the well, and the well coverage parameter shown here indicates the size of this black circle that is going to be the part of the well that is covered by your images. The field density, the second parameter, says for each of these little yellow rectangles that represents the size of one image inside the well, inside the plate, how close do I want them? If you want them close together or overlapping, then the field density would be 100% or higher. If you want a sparse sampling across the plate, you would use a lower number, and then you'll get empty spaces that are not imaged between each field of view, each image area right here. Next, you can optionally define your time lapse if you have one. If not, you do just a single cycle, which is to image the plate one time. Another set of options that you can input is dataset information. So this is a free text box where you can type whatever you want. Tags is the way that you can upload concentration and treatment conditions per well. So you can say which compound you're using in each well and what's the concentration in order to extract a dose curve at the end of the analysis. This is optional here before acquisition. It can also be uploaded into Athena before image analysis. Optionally, you can set positive and negative controls. And from the default settings, you're given an estimated time for the scan to take place. Once you're done here, click on Done, and then you're going to enter into the scanner screen. Once you're in here, for each color channel that you select, in this case it's blue, green, and red, then you're going to get a readout of the image here. You can look at an overlay still image here. And for each color channel, you define the focus plane, the light intensity, the exposure, the camera gain, and that's it, right? Once you set these power and detection parameters and the focus plane, then you just click Done. You go to the summary screen, you'll get a review of what you're going to do, and you begin the acquisition, right? So just 11 steps to go from loading a plate to getting your images, beginning their acquisition steps.